students, my name is Dr. Salahuddin, working as Associate Professor in Noida Institute of Engineering and Technology, Greater Noida, Pharmacy Institute. And today, I will discuss about the combinatorial chemistry. So, in the combinatorial chemistry, basically we are synthesizing the compounds in a very specific manner. In the combinatorial chemistry, that we can apply the either the solid phase or the liquid phase synthesis or the solution phase synthesis. So, first is it is developed in 1980s with Giesens and Huntanks. So, the basic principle of the combinatorial chemistry is what? It is to prepare a large number of the similar compound, large number of similar compound that is not applicable for the different type of compounds. So, here there is a limitation, that is large number of the similar compounds at the same time. Instead of synthesizing compounds in the conventional one at a time. So, here in the conventional synthesis, that we are synthesizing a compounds one by one. If I am searching a new ligand, a new pharmacophore, that we have to synthesize the compound in our lab one by one. So, it is a conventional synthesis, but with the help of combinatorial chemistry, we can synthesize the number of compounds, a large number of the derivatives at one time. So, a large number of the compounds will be produced. So, the basic principle involved here, that is it is a preparation of the large number of compounds at the same time, instead of synthesizing the compounds it is one by one, that is the conventional synthesis. So, the objective, our objective is what? Our objective is to prepare the library of the compounds, so that we can opt this the combinatorial chemistry. So, in the combinatorial chemistry, we can build a large library of the compound. Library means the collection of the compound at one place from a starting, that is a scaffold. So, when we are synthesizing the compounds, not the compounds synthesized in one step, we have to apply the step by step synthesis. So, we have to choose the scaffold, choose the simple structure to interact with the specific biological target, that is the combinatorial library, the collection of the finally synthesized molecule in the combinatorial chemistry. So, the combinatorial chemistry involves the generation of a large array of the structurally diverse compounds called a chemical library through a systemic, repetitive and the covalent linkage of various building blocks. So, this is the very important that we can use the various building blocks, and with the help of these building blocks, that we can prepare a number of compounds, that we can evolve a generation of a large structurally diverse the compounds by using these simple building blocks. So, the compounds in the chemical library that we can screen. Nowadays, the various libraries are available, online libraries, but here here we are synthesizing the compounds, synthesizing with the help of the combinatorial chemistry. So, this combinatorial chemistry helps to produce the library, and after the formation of the compound, that we can screen all these compounds for the individual interactions with the biological target of our interest. Here the types of combinatorial libraries in the drug design, that we have the two libraries, random library and focused or targeted library. Focused or targeted library. Random library, the multiple libraries, and this is based on the template and the scaffold libraries. So, what is this template? The similar type of template, the similar type of scaffold that we can access from these focused or or targeted libraries. Here there are many targets are involved in the random library, but here because it is a focused or target library, it is one target and it is highly diverse compound, it is scattered everywhere. So, here high structural similarities that we can choose from this focused or targeted libraries, that is high structural similarity that we can obtain with the help of this focused or targeted libraries. Generally, here in the random libraries more than 5,000 of the compounds. In the focused or targeted libraries, we already filtered the compounds. So, here this is very less than the 5,000 of the compounds. Here it may be mixture of the compounds and here we can get a single compound. Here the non purified compounds, here we can pure compounds. So, our main aim here the main aim is to produce the focused or the targeted libraries that it can be possible with the help of this combinatorial chemistry. Now, this combinatorial chemistry within the drug design, where this combinatorial chemistry involved in the drug discovery process. So, in the drug discovery process, first thing is the therapeutic target, we have the therapeutic target. So, from where we can obtain this therapeutic target? If the disease is new, that we can isolate the target. This is another a very critical step for the isolation of the target. So, here the therapeutic target nowadays there is a various types of target available online, that and against this target we have to find out the lead compound. So, find out the lead compound is very important step in the drug discovery. Finding the lead compounds with the help of combinatorial chemistry. So, first of all that we have to determine the basic scaffold with the help of the lead discovery, and then we can synthesize the large number of diverse derivatives that it can be done with the help of this combinatorial chemistry. So, here this drug, when the drug discovery the lead discovery will be done, and this after the lead discovery we have to do the lead optimization. So, here at this step, from the lead discovery to the lead optimization, we have to apply for the combinatorial chemistry to generate the various types of the compounds, to generate the library, the diverse compounds, and we can screen all these compounds with the help of high throughput screening, another branch, that is in vitro screening. And after the screening, we will get this development candidate, and this development candidate may be converted into the drug. So, our aim is here to synthesize the number of compounds by using this combinatorial chemistry. Now, in this combinatorial chemistry, in the drug design, so first thing is identification of the drug target and the development of assay, high throughput screening. From this high throughput screening, we will get the hit compound, and this hit may be converted into the lead structure, and this lead optimization and development of this development of new candidate. So, here I have already discussed this random libraries and the focused libraries. So, from this random libraries, we can perform this high throughput screening and this focused libraries. From this random libraries, we can convert into the focused libraries by applying this combinatorial chemistry. So, after applying this combinatorial chemistry, we can develop this focused libraries, and from this focused libraries, the hit or lead structure that we can obtain in the drug design. So, the combinatorial chemistry, it is a lead drug discovery in the traditional lead drug discovery, that we were found the compound from the natural product, synthetic custom crafted organic molecule made in small numbers, analogues of the known actives, that is analog me too drugs. Now, high throughput screening required a large number of compounds for the screening for the activity, and as an alternative to the traditional synthetic synthesis, may compound rapidly constructed was needed. So, here the various tools nowadays is available. In the combinatorial chemistry, basically there are two basic tools are available, or the two basic methods that we can apply the synthesis of the compounds. In the synthesis of compounds, that we can apply this solid phase synthesis, and in the solid phase synthesis there are some different methods are also available. T-bag synthesis for split and mix synthesis, multi-pin synthesis, and this the next one is solution phase synthesis. So, in this lecture I will discuss about the solid phase synthesis first. Now, in the solid phase synthesis, what is happening? So, as we know that in the synthesis of the compounds, we require the reactants. So, the reactant first it bounds with the polymeric surface and modifies still attached. The final product is released at the end of the synthesis. So, in the solid phase synthesis, our reactant bounds with this polymeric surface at the reaction condition. So, after the modification of the compounds, or then we have to remove this polymer or remove this polymeric surface, and after that at the final step we will get the compounds. So, the advantage is what of this solid phase synthesis? A specific reactant, and it can be bound to a specific beads. So, what is beads? So, here beads can be polymeric surface. The beads can be mixed and reacted in a same reaction vessel. So, here we can use the same reaction vessel that we have the polymeric surface, that is bed also, and the reactant also. There is no separate vessel that we are using. So, it can be mixed and reacted in the same reaction vessel. So, after the reaction, the product will be formed. So, product formed are distinctive for each bed and physically distinct. Now, next thing is excess reagent can be used to drive the reaction to the completion. Here the excess reagents and the byproducts formed in the reaction. Whenever we are doing any type of reaction, there is a possibility to form the byproduct. So, we have to remove this byproduct, and we are using here this polymeric surface or bed, that we have to remove this polymeric surface also. So, excess the reagents, unreacted reagents and the byproducts we can easily remove with the help of this solid phase techniques. Next thing is reaction intermediates are attached to the bed and do not need to be isolated and purified. Individual bed can be separated to isolate the individual product. So, here the polymeric support can be regenerated and reused after cleaving the product. So, whatever the polymeric support that we are using, so after the cleaving, after the isolation of the product, that we can reuse these polymeric support. So, what is the requirement? Requirement in the solid phase synthesis. So, here the first thing that we require a resin bed. Or a functionalized surface to act as a solid support. This resin bed is the necessary step. An anchor or a linker. So, anchor or linker, why we are using this anchor or linker? So, the basic function of this anchor or linker, this anchor or linker have two ends. So, at one end it will interact with the solid support, that is the polymeric surface. At the different end, another end will react with the reagent to form a intermediate. So, a bond linking the substrate to the linker, the bond must be stable to the reaction condition used in the synthesis. Whatever the bond formation, with the help of this linker, the bonds must be stable, otherwise it will break into the some other product also. A means of the cleaving of the product from the linker at the end. So, here we are using the resin bed or the linker and cleaving of the product from the linker and the end. Whatever the product formed in this reaction, and in this reaction that we are using the resin bed, anchor or linker. So, we have to remove the anchor or linker at the end when the reaction completed. Now, next thing is protecting group. So, the protecting group for the functional groups not involved in the synthesis. At sometimes we require the protecting group in the reaction to protect the some functional group that the not involved in the reaction. Now, here the example of the solid support. Some example of the solid support. So, the solid support are partially cross linked polystyrene bed, hydrophobic in nature, water heating, causes problem in the peptide synthesis due to the peptide folding. Next is Schaeffer's polyamide resin, and this Schaeffer's polyamide resin is more polar, and there is that we can use in the as a solid support. Tentagel resin, that is the similar environment to other or THF, that is beds, pins or the functionized glass surface that we can use as a solid support. So, what are the characteristics of the solid support? So, the beds must be able to swell in the solvent. So, whatever that we are using as a polymeric bed, polymeric support for the completion of the reaction, that have a specific characteristic, it must be able to swell in the solvent and remain stable, not to degrade it into the some other product. So, most reaction occurs in bed interior. Here there is a resin bed in the form of this blue color. So, after swelling that after swelling it will swells, and here the starting and material and the solvent that in the form of that linker that we can use. As I have already told that linker have two ends. So, here a molecular moiety which is a covalent attached to the solid support, which contains a reactive functional group, allows the attachment of the first reactant. First it will interact with the solid support and the first attachment with the reactant. So, the linker or anchor both are the same. The link must be stable. The polymeric support, that is also stable at the solvent condition. Here the anchor or linker, that is also must be stable to the reaction condition, that whenever we are applying the heat or some other condition, it must not be degraded into the other. So, it must be stable to the reaction condition in the synthesis, but easily cleave to release the final product in a pure form. Different linkers are available depending upon the functional group to be attached. Our requirement is different, different types of linker will be required for the different type of the reaction, different type of the product. If the product is different, so the linker must be different. So, desired functional group, it is dependent upon the desired functional group on the product. Resins are named to define the linker, that is Merrifield resin, Wang resin and the Ling resin. In the solid phase synthesis, here this is the resin and this is the linker, and at one end it will link with this resin, and there are some groups that that is, and this will form with the reactant. So, here it will here there is using this Wang resin linker suitable for the attachment of release of the carboxylic acid, the Merrifield resin linker suitable for the peptide products, Rink resin, that is the linker suitable for the attachment of release of the carboxamide. So, here the different types of the resin that we can use for the different types of reaction, as here for the carboxylic acid is different, for the peptide product is different, carboxamide, that is a polymeric resin, that is the solid support is different. So, the solid phase synthesis, there are various types of protecting groups that we are using. For amines, if our product having the amines, so here the protecting groups that we are using, that is BOC, tertiary butoxy carbonyl, FMOC 9 fluoro ethylene methoxy carbonyl, that is NIMSAC, that is 2 trimethyl silyl ethoxy carbonyl, and for the carboxylic acid it is also different, tertiary butyl ester, 9 fluoryl methyl ester, 2 fluoryl methyl silyl ethyl. So, the solid phase synthesis, the various types of functional group that we are using. Here this is the example of the Merrifield resin for the peptide synthesis. Here we have the resin bed, and this is the linker, and the various types of here also we are using this protecting group. So, after the protecting group that we are using to protect the some specific functional group, and we can get the final product after removing this protecting group also. So, this in the solid phase synthesis, the advantage is synthetic intermediates do not have to be isolated, it is a quick process, reagent simply washed away each step. The disadvantage also, all the synthesis can't be done on the solid phase, typically kinetic not the same, unsuitable for the solvent assisted chemical reaction, high viscosity in the reactant system, insufficient purity of the reaction steps are incomplete. And lastly, for the solid phase synthesis, R. Bruce Merrifield given the won the Nobel Prize in the chemistry in the 1984. These are the some references. So, thank you very much.